So, as you all know, the gastrointestinal ecosystem has a tremendous influence on our overall health and well-being, as well as our ability to detoxify. And as early as 400 BC, Hippocrates, kind of a radical, stated that death sits in the bowels and bad digestion is the root of all evil. Now recently, the gastrointestinal metabolome, or microbiome project, has cataloged the overwhelming number of unique genetic sequences that encode for the phenomenal number of gastrointestinal bacteria. Now someday that genetic information will be clinically manageable, but I've spoken with several world-renowned microbiologists that have stated that the biggest thing that we have learned from that project, the genetic project, to date, is just how little we really do know about the gastrointestinal metabolome. Okay, so genes are one thing, metabolism is another. So the genetic information has absolutely been in the spotlight at the expense of the metabolome, especially regarding the interactions between the microbial metabolism and the integrity of the essential intestinal barrier system. So today we're going to focus on the complex gastrointestinal barrier systems in the context of excluding toxic, antigenic, and pro-inflammatory macromolecules from the lumen that can affect every organ system in the body. And my sincere hope is that this discussion will help you more thoroughly appreciate and act upon some often glossed over results from your favorite comprehensive stool analysis. Okay. So, full disclosure, as Dr. Park said, I do work for Doctor's Data, and I don't make stuff up. All of my citations are level C or higher. So when we think about the gastrointestinal ecosystem, we have to consider it like any other ecosystem in the world, okay? And that is, it entails a network of interactions among and between the organisms, and very importantly, among and with the environment. And in this case, the environment is shown by the frame drawn around this beautiful coral scene here, and that represents the gastrointestinal barrier system. So we have this incredible communication between the microbes themselves, as well as between the microbes and the intestinal barrier system, which I will describe in great detail. It's an incredible symbiotic relationship where the microbes support the barrier, and the barrier supports, protects, and helps out the microbes. It's a really, really slick system. And back in the 1800s, Claude Bernard said that the germ is nothing. Well, bacteria are certainly something. But the point to take home is the terrain is everything. And by the terrain, I'm talking about this picture frame around this picture here that is the intestinal barrier system. So we're really going to focus on the terrain containing the microbes. So the GI ecosystem is composed of the metabolome, that is the microbes, their genetic material, plus their collective metabolites. And then we have the terrain, which are the intestinal barriers with an S. Remember, it's not just the endothelial cells. And then also, very importantly, we have the gastrointestinal glycobiome, another OM for you to take home. And glycomics is simply the study of glycosylation of molecules. And in the gut, the most important molecules being glycosylated are the mucins, which are highly glycosylated proteins that are constantly renewed. They are the primary molecules involved with host pathogen interactions. And we have both secretory and adherent mucins, which are both critical for the protective barrier layers. Now, these mucins are extremely dynamic. It's almost like they're alive. They're glycoproteins, but it's almost like they're alive because they're very responsive to changes in conditions in the gut, including diet, different microbes, and how the immune system is affecting the gut. And to give you an example of how dynamic these mucins are, consider that during infectious invasions, the distinct mucin glycans of both the host and the pathogens change, which can affect pathogenesis as well as the host structural and immune responses. So the mucins are constantly changing depending on what's going on. So I'd like to give you an idea of where I'm going and then summarize what I've told you so that hopefully things stick better. So what I'm going to talk about is the primary components of the intestinal barriers, the key regulatory factors, including the severely underappreciated commensal Clostridium species. We're going to look at indirect assessment of barrier integrity from a comprehensive stool analysis, looking at things like butyrate, secretory IgA, and inflammation. And then we're going to get deep and look at direct assessment of that final barrier, the endothelial cell barrier, by looking at a regulator of the tight junction protein complexes. That would be zonulin. And everything that you're here for, of course, we're going to talk about therapeutic support for barrier integrity. Now, keep in mind when we talk about intestinal permeability, we have both transcellular through the cells as well as paracellular between the gaps between the cells that shouldn't be there. So we're really going to dive in deep and delineate the zonulin pathway and talk about consequences of sustained dysfunction of that final epithelial barrier. So we can have breaches in the gastrointestinal barriers induced by diet, caloric deprivation breaks down the mucin barriers, a low fiber diet doesn't supply enough of the mucin. We can have breaches due to changes in the microbiota and the metabolome, and we're going to see that with insufficiency dysbiosis. Infection, poor lifestyle choices, including excessive alcohol, overuse of pharmaceuticals, obviously antibiotics, prednisone, and NSAIDs. Stress is a huge factor on the intestinal barrier. Gut-derived lipopolysaccharides, otherwise known as the O antigen, and undigested protein fragments, direct adhesion of bacteria to endothelial cells. It really ticks them off. They don't like it at all. And it really disrupts the whole system. Obviously, environmental xenobiotics, inflammation, direct epithelial damage, cancer treatments are very hard on the gut, and excessive exercise, particularly with heat stress. Not your normal exercise, but excessive, gross exercise. So the intestinal barrier system is a phenomenal, multifactorial, layered, and highly integrated system. It's a very, very high maintenance system. Much more high maintenance than my first wife. It takes about 40% of my 40% of my BMR. So it's a very expensive process to maintain. Primary functions of the intestinal barrier system, of course, are for water and electrolyte balance, simple stuff, but important. But also, very importantly, to prevent the influx of microbes, toxicants, and antigens from the lumen of the gut. And the barrier system regulates appropriate inflammatory and immune responses. Remember, not all inflammation is bad. So we can have tolerance or a robust immune response. And this intestinal barrier system microbial relationship is an amazing two-way street. there is mutual regulation of the microbes and the barriers. That is, the commensal bacteria, otherwise known as beneficial bacteria, facilitate optimal status of the barrier components, and in turn, the barrier components provide surveillance, protection, and selection of the commensal bacteria and eradication and elimination of the pathogenic bacteria. Now, it's truly a multi-layered system that is composed of the commensal bacteria and their metabolites. Then we have a functional chemical or biochemical barrier that includes digestive enzymes, immune molecules such as secretory IgA, antimicrobial peptides, and inflammatory mediators such as the cytokines. And then finally, we have the actual physical external barrier that is composed of the mucus gradient, the glycocalyx, over top of the epithelial cells with their tight junctions, that sits on top of the lamina propria, and then finally the vascular endothelium. So it's not just endothelial cells sitting there naked. And unfortunately, the term leaky gut is out there, and it's a term that drives me nuts. But typically when people are talking about leaky gut, they're talking about breaching that final barrier, the epithelial cell barrier, by breakdown of the tight junction proteins. But there's so much more to the story than that. So to kind of put this into a clinical perspective, we'll look at gross insufficiency dysbiosis that was coined many years ago, I believe, by Dr. Leo Gallen. And this is a case of a 22-year-old female who unfortunately was put on broad-spectrum antibiotics for a bad cold. Okay, go to your doctor, you have a cold, here's a prescription. Well, obviously it knocked out her beneficial bacteria. She didn't have any GI symptoms, but she shortly thereafter developed arthritic-like pain and inflammation in her hands, a 22-year-old female. And as you can see, her beneficial gills, the commensal bacteria, were knocked out by the broad-spectrum antibiotics. There was no growth of bifidobacter, marginal growth of E. coli, no growth of lactobacillus, poor growth of Enterococcus species, and marginal growth of the commensal Clostridium species. When this happens, we have a vacancy of adhesion sites, and a lot of opportunistic bacteria can move in and colonize, and those are shown in the yellow portion on this chart. Now, one doesn't directly treat those opportunistic bacteria. Most of them are very benign. Some have some negative properties, but some may even have redundant beneficial properties. The point is, it's a very strong validation that you have a disrupted microbiome, okay? So what happens when we have a vacancy of these commensal bacteria, we compromise our intestinal barriers. And with loss of those key commensal gills, we get a marked reduction in butyrate, which is the primary signaling messenger for the microbial host crosstalk. It regulates the production and secretion of secretory IgA, antimicrobial peptides, mucus and mucin, and the overall effect is a breakdown of our mucus and mucosal barriers and increased permeability. Now keep in mind when that happens, it doesn't only affect what's going on in the gut, but it can also result in systemic inflammation and even systemic autoimmune conditions. Now, I'm a very visual person, and I kind of want to put this into a picture form so it sticks into your head more. We start out close to the endothelial cells at the bottom there, where the goblet cells secrete mucin. And as we go up towards the core or the center of the lumen, we get a breakdown of the inner firm mucin by protease activity. And that forms an outer loose river of mucus. And that's where the pathogens originally enter, and with normal bowel transit time and peristalsis, it helps move, that river helps move the pathogens out of the body. Then the second component of that mucus blanket is the inner firm layer, very, very dense layer of mucus and mucins, and that's what houses the commensal bacteria, secretory IgA, antimicrobial peptides, reactive oxygen species, and others. Then underneath that dense mucus layer, we have another non-secretory mucin layer called the glycocalyx, which is directly tethered to the epithelial cells. Then we have the classic mucosa, where we have the epithelial cells, their tight junctions, and their recycling apical endosomes. And then the fibrous tissue, the lamina propria, that contains those juicy dendrocytes, T cells, B cells, and lymphocytes. So there's a whole lot more above the endothelial cell barrier that we need to think about. And to drive home that point, just consider that from the literature, it's been demonstrated that ulcerative colitis patients have a disrupted microbiome, decreased mucus thickness, increased pathogen colonization, and permeability. So that's how important the slime layer is. Now, there's specialized cells in the mucosa that are responsible for sensing, reporting, and secreting. On the far left, we have the enterocytes that secrete hydrogen peroxide as an antimicrobial. Then we have the Paneth cells through the toll-like 2 receptors secrete antimicrobial peptides and the glycoside hydrolytic protein enzyme lysozyme. Then, of course, we have the B cells producing IgA that is dimerized to form secretory IgA. And then 2 of my favorite cells, in the center there in blue is the goblet cell. And the goblet cells remind me sort of like blowfish, because they produce mass quantities of mucin, which is basically mucus. And they have little mouths like a blowfish, and that's how they release their mucins. And when their mouths are open, antigens can get in. And the antigens are transported through the goblet cells and down and communicate with associated dendritic cells in the immune system. The dendritic cells say, hey, blowfish, blow your wad, shoot this antigen stuff out of here. Get rid of it. So it's really a slick system between the immune system and these cells. And then my very most favorite cells are the dendritic cells. These are what I call the long arm of the law of the lamina propria, because these dendritic cells have these phenomenal dendritic feet that reach up through these specialized M cells and actually grab bacterium off the surface of the glycocalyx, bring it down, present it to the immune system for a yay or a nay. So we have this phenomenal system of finely tuned network of immune mechanisms for microbial recognition, selection, and eradication. Now, I've been presenting this as a linear system, and you all know that there's a phenomenal surface area. It's not linear at all because of the villi and the microvilli. And then when you look at a blow-up of the specialized columnar-like endothelial cells, the enterocytes, you can see that they're very tight together, and they have these little fuzzy hairdos. And anytime I look at one of these in an electron micrograph or in a diagram like this, I can't help but think of one of my favorite cartoon characters, because he has that columnar-shaped head. He's got that sporty Villus-like hairdo that can only be formed by applying a viscous gel, which would be reminiscent of the glycocalyx. He's got those beady eyes and that nose for sensing, and his big mouth for reporting. And then if you really use your imagination, remember his little itty-bitty ears? Then you might imagine those to be the tight junction proteins that hold those enterocytes together. So next time you see Mr. Bart, you might think of an enterocyte. Now, mucins are a critical component of maintaining the barrier function. They're highly glycosylated proteins that form a polymeric gel-like network. They have sulfhydryl bonds that form a net structure, and because of the high degree of glycosylation, it attracts a lot of water. So it's like a spongy pillow with very, very small pores that don't allow things as big as bacteria to get through. So we have secretory mucins, such as MUC2 is a famous one if you're looking in the literature, which serves as a mucus barrier and also as a habitat and nourishment for those commensal bacterial guilds. Then we have the non-secretory cell-bound mucins, which compose the glycocalyx. They serve as binding sites and also as a barrier, and they also signal to the enterocytes. So they prevent direct binding of any bacteria. The endothelial cells don't even like lactobacillus binding to them. It really ticks them off. And so this is a barrier to protect these endothelial cells. And interestingly, I was reading a paper about, and I don't know whether this has actually been demonstrated or just a theory, that these non-secretory glycocalyx mucins can serve as decoys. And that is depicted here as you see these three different mucin molecules with their fuzzy green glycan portions and the red protein transmembrane tail. And it's been suggested, probably from evidence, that when pathogenic bacteria bind to the surface of these mucins, that it signals to the enterocytes, and the enterocytes signal the immune system, and it signals back, and they actually release the antigen back up into the layer that contains the antimicrobial peptides, the secretory IgA, and all that good weaponry. So, decoys. So the mucins are regulated by commensal bacteria within the mucus. This is wrong here, it says mucosa, but within the mucus versus the lumen. Okay, and so what we have is mucin harvesting specialist bacteria that break down and release the glycans from these proteoglycans. Those glycans are then fermented by the commensal bacteria to produce butyrate, and that butyrate signals for more mucin production. So it's a really nice cycle. And so the mucins are to the endothelial cells as biofilm is to bacteria. Very protective, okay? And all bacteria and all yeast have biofilm. Endothelial cells really, really need to have this viscous glycocalyx coating. Now, with obesity and high saturated fat diets, we see decreased mucus thickness and increased inflammation and increased permeability of the mucus barrier. On the other hand, using probiotics, which are the soluble oligosaccharides, you can obtain them from many different kinds of foods. They actually increase the mucin-stimulating bacteria. And those foods include onions, garlic, Brussels sprouts, asparagus, leeks, all those foods kids love to eat, right? Yams, chicory root, agave, bananas, and the Jerusalem-type artichoke. So just to recap the microbial host mucin cycle, just because it's so important, the mucins are the host prebiotic. The protective mucus layer at the epithelial surface is continuously reshaped and refreshed, about 5 liters a day. Most of you don't drink anywhere near that much water. That's how much work these little blowfish goblet cells are doing, 5 liters a day. It's amazing. So its glycoproteins harbor and feed the symbionts. And if you have a low fiber diet or starvation, and we see this with anorexia nervosa, the beneficial bacteria just gobble up the mucins, and you get a decrease in the mucin barrier. So we have a consortium of mucin-degrading specialists within the inner mucus layer that release the glycans that are fermented by other commensal bacterial guilds that crank out the butyrate, and that butyrate crosstalk stimulates mucin production and regulates immune function, inflammation, and basically fortifies the mucus barrier matrix. Now, a grossly misunderstood genus of bacteria are the Clostridium. Yes, there are 5 truly pathogenic Clostridia, but many people are walking around even with toxin A and B producing C. difficile. As long as you have good commensal bacteria, you're not going to get C. difficile associated disease. But there are about 100 different species of commensal Clostridia. People hear Clostridia and they freak out. They think it's a hospital-acquired disease. It's actually rampant in the community. There's people walking around with C. diff, and it's not a problem. But the important point I want to make is that about 5% of the total bacteria in that strategic position above the glycocalyx are actually Clostridium species, and they are major butyrate producers. And the commensal, they include the Clostridium histolyticum, Lytospirans, and cluster 14A. So they're integral symbionts in the regulation of barrier homeostasis. We see a decreased abundance of commensal Clostridium species with colorectal cancer and IBD. Okay, and there's another clue that it's important. On the other hand, we see an abundance of Clostridium species, commensal, in association with consumption of high fiber intake from fruits, vegetables, beans, and legumes. Now, if I haven't convinced you that Clostridia can be good guys, at this point, consider this, that during the first month of life, one of the primary species of bacteria or genuses of bacteria present in the breastfed infant's gut are Clostridia. They colonize the infant's gut within the first month of life, that is in breastfed infants. And it's really a cool way that this happens. It occurs by vertical transfer from mom's gut to her milk to the infant. And this was demonstrated very eloquently in a human study of 7 moms and their infants. And what they did was microbial analysis of mom's breast milk and of maternal and infant feces. And they used 16S ribosomal RNA pyrosequencing, not just PCR. So, as expected, they found an abundance of bifidobacterium, but also an abundance of Clostridium bacterium, including C. blauti, Collencella, and Veillonella. So what you have is Clostridia from mom's gut, taken up into her lymph, put into her breast milk, and then into the infant's gut. And the mechanism is my favorite cells, the subendothelial dendrocytes, the dendritic feet, that actually sample and transport mom's gut bacteria into her lymph. And so depicted here again is that those long dendritic feet reaching up through those M cells, snatching the Clostridium, bringing it down. The immune system says, cool, put it in the lymph, and goes off to the breast milk and ends up in the infant. So, another major regulatory factor and certainly a major metabolite of the commensal bacterial guilds are the short-chain fatty acids. And the short-chain fatty acids, as you all know, are fermented from the mucin-derived glycans, as well as from soluble dietary fiber. A lot of people think we only get short-chain fatty acids from what we eat. But again, think about breaking down those mucins. So butyrate mediates microbial host crosstalk. That is, the host actually listens to butyrate. And that occurs through G-coupled protein receptors on the surface of immune cells and epithelial cells. So the butyrate mediates production of mucins, antimicrobial peptides, and the release of anti-inflammatory cytokines by the epithelial cells. The butyrate also regulates expression of the tight junction proteins between the endothelial cells, gene expression. Also, butyrate is rapidly taken up into circulation. You don't measure butyrate, you can't measure butyrate levels. It's cleared very rapidly, that which is taken up systemically. In fact, it crosses the blood-brain barrier and can significantly ameliorate neuroinflammation. So there's another part of the gut-brain connection. Neuroinflammation is a huge issue. Faecalibacterium prausnitzii and some other Clostridia are major butyrate producers in that strategic real estate in the barrier system. And these bacterium are much more abundant in diets containing chickpeas and raffinose. Now, raffinose is a trisaccharide consisting of galactose, fructose, and glucose. And we lack the enzyme alpha-galactosidase to break down this trisaccharide. And my friend from England refers to this as the sugar that causes wind. Okay? And so raffinose and stachyose are soluble saccharides that make it all the way down to our beneficial bacteria. They ferment it to produce the butyrate, which, if you can't put up with a little wind and you use the Beano all the time, Beano is actually a product that contains the alpha-galactosidase. And no, I have no vested interest in Beano. Okay? Now, another major component, and it's what I call the brick in the mucus barrier, is secretory IgA. It's anchored in the dense, viscous mucus blanket and on the epithelial cells. It provides immune exclusion of microbes, including viruses and toxins. And the secretory IgA binds to the pathogens, binds to their antigenic epitopes, and it causes a decreased diffusion of the pathogens. It changes the membrane composition, which results in decreased ATP production of the pathogens, which decreases their ability to be motile and decreases their ability to form their protective biofilm. So secretory IgA is doing a lot of stuff. It's also anti-inflammatory. It neutralizes the lipopolysaccharides in the apical recycling endosomes. So it abrogates that NF-kappa B pro-inflammatory signaling. It also augments antiparasitic activity by activating eosinophils. It regulates the composition of the microbiota, providing constant surveillance, sampling, and communication, like the dendrocytes, with the immune system. Now, with chronic candidiasis, it's often associated with extremely low levels of secretory IgA. And that's because in a study where they took the Candida species from 38 different Candida species from people with yeast overgrowth, those 38 species secreted a secretory IgA specific protease. So the yeast have their own weapons to tear down the major brick in our mucosal barrier. Now, we should see increased secretory IgA in a stool sample if an enteropathogen is present. If you don't see dysbiotic bacteria or parasites or yeast, and secretory IgA is elevated, think of a recent viral infection, because secretory IgA will remain elevated for 4 to 6 weeks after an acute gastritis due to a virus. Okay. Intervention for low secretory IgA, nobody should have low secretory IgA. You can use omega-3 fatty acids, short-chain fatty acids, olive oil, zinc, D3, vitamin A, probiotics such as Lactobacillus rhamnosus GG, which is the most heavily studied probiotic in the world, as well as Bifidobacterium lactis BB12. Both have been shown to increase the number of secretory IgA producing cells in formula-fed infants. And then you're all familiar with Saccharomyces boulardii ability to increase secretory IgA, and that's been demonstrated in germ-free mouse models. You can also use prebiotics from some of the vegetables, fruits, chickpeas, beans, etc., glutamine, arginine. But on the other side of the coin, we know that stress knocks out secretory IgA. So if your patient has no dysbiotic flora, no dysbiotic yeast, and secretory IgA is low, consider cortisol and DHEA. Okay. So I want to say just a couple more things about vitamin D, because it's so vital, and all your patients, you really should evaluate their vitamin D status. It's important for so many things. And vitamin D receptor signaling through the VDR, vitamin D receptor, turns off chronically activated T cells. It decreases T cell proliferation and their inflammatory cytokine production, thereby decreasing mucosal inflammation. Vitamin D also enhances the expression of those endothelial cell tight junction proteins. It influences the microbiome and barrier integrity by regulation of antimicrobial peptides as well, and basically facilitates colonic cell healing and overall barrier function. So vitamin D is a very important component to have in your quiver when addressing patients with GI issues and systemic issues as well. So there are two primary ways that we can breach the final barrier, the final frontier, the furthest away from the lumen, the epithelial cell barrier. We can breach it normally through transcellular transport that is typically mediated by endocytosis or receptor-specific mechanisms. So things like electrolytes, B12, amino acids, sugars, short-chain fatty acids, and even secretory IgA can be transported directly through the intact cell. Also, a small amount of antigens and bacterial translocation can occur. But then we have paracellular transport, which is unregulated passive diffusion when we have a breakdown of those tight junction proteins between the epithelial cells. So depicted here is a normal mucus layer where we have an abundance of secretory IgA and commensal bacteria. Yes, there's some transcellular transport and very minimal transport through the tight junction proteins, such that it just keeps the immune system on its toes and you have tolerance. However, when you have an injured or damaged mucus layer, a thinned mucus layer, there's less secretory IgA, there's less beneficial bacteria, commensal bacteria, and more pathogenic bacteria. That increases both the transcellular bacterial translocation as well as the paracellular transport through the broken tight junction proteins. And this results in a very undesirable response that I refer to as the crazed immune response. So that final cellular barrier is the epithelial cell tight junctions, and it is broken down by a protein called zonulin. And zonulin is responsible for increased permeability of the epithelial cells. Now, those tight junctions are dynamic, interconnected protein networks that regulate paracellular influx for all epithelial cells. That's not only in the gut, but also the epithelial cells in the vascular lining in the heart, as well as in the blood-brain barrier. And zonulin protein is currently the only known physiologically reversible, keyword is reversible, modulator of those intracellular tight junction protein complexes. Now, reversible is a key word because we can have transient reversible opening of those tight junctions. That's a normal physiological process. And when I first started reading about this, I thought, well, why in the heck would the body do that? Why would it open the gates like that? Well, then as I continued to read the work of Dr. Fasano, who's the guru of celiac and zonulin, his theory is that there is transient opening and closing of the tight junctions, very transient, to provide an influx of toxins and bacteria that facilitates a defense mechanism. So you let in some bacteria that are undesirable, and that provides a robust immune response. And that makes sense because it doesn't stay open. The problem is when we have chronic, sustained opening of those tight junction proteins, and that occurs with high serum zonulin levels, and that's associated with autoimmune diseases such as celiac, Crohn's, rheumatoid arthritis, type 1 diabetes, lupus, lichen planus, cancers even, gliomas, breast cancer, ovarian, pancreatic, and neurological diseases such as demyelinating polyneuropathy, multiple sclerosis, another autoimmune disease, schizophrenia, and non-celiac gluten sensitivity, as well as the pandemic juvenile fatty liver disease in North America, including our friends in Canada, and asthma and metabolic syndrome. So again, being a very visual person, I want to leave you with this picture of how gliadin induces zonulin release, increases intestinal permeability, and causes the onset of autoimmunity. Gliadin binds to a specific receptor called the CXCR3 chemokine receptor, and then through assistance of some adapter proteins and molecules like MyD88, it actually causes the ribosomes, or the endoplasmic reticulum rather, to release an abundance of zonulin. And in active celiac disease, where they're exposed to gluten, the CXCR3 receptors and zonulin are overexpressed. So the endoplasmic reticulum releases the zonulin from inside the enterocyte out through the apical surface, where it binds and transactivates two very important receptors, the protein activating receptor 2 and the epidermal growth factor receptor. Activating those receptors turns on a proteolytic cascade that tears apart the tight junction proteins. And then we have unregulated influx of highly antigenic, pro-inflammatory molecules that just really charge up the immune system. And the immune system not only attacks the cells in the gastrointestinal tract, but also specifically attacks the beta cells in the pancreas, which is where we see type 1 diabetes. So increased permeability of the gut can be associated with type 1 diabetes. So gluten sensitivity, a lot of stuff, and it depends on what you read and where you read. it's not just a fad. I have severe gluten sensitivity, very, very rapid onset. And talking with my 89-year-old father, he just doesn't buy it. He said, "Eat the damn bread." You know, no, Pop, you don't want me to eat the bread, trust me, okay? But even in the 2015 article from Gastroenterology and on the Mayo Clinic website, they have stated that when people with GI diseases such as celiac, non-celiac gluten sensitivity, or even IBS consume gluten proteins, they can become sick. Now, no doubt, there are some people that are on the fad bandwagon, but I want to show you some data to show that it's not really just a fad. This is showing the serum zonulin levels on the y-axis across patients or subjects with different diseases. Celiac disease has the highest level of zonulin, as would be expected. Slightly lower, but statistically higher than healthy controls, are non-celiac gluten-sensitive individuals. But not different than controls were the levels of zonulin in IBS patients versus the healthy controls. So this was an interesting initial study that was followed up by a very definitive study that showed a very good response of decreased serum zonulin and permeability when certain patients were put on a gluten-free diet. Now, zonulin, that is the serum antigen measured in blood and from a blood sample, levels were assessed in patients with celiac, non-celiac gluten sensitivity, IBS, and IBDs before and after being on a gluten-free diet. And as anticipated, the patients with organic significant gastrointestinal inflammation, there was no effect of the gluten-free diet in the IBD patients. They maintained their elevated levels of serum zonulin. However, in the celiac patients, just like we would expect, the gluten-free diet resulted in a significantly lower serum zonulin and antibody titers for the biomarkers of that extreme tissue damage, the tissue transglutaminase and the deaminated gliadin peptide. Now, in non-celiac gluten-sensitive patients, there was a decrease in zonulin and a decrease in IgG and IgA for gliadin antibodies. Now, interestingly, for the IBS type D patients, there was a greater decrease in zonulin in the subjects that had the double HLA-DQ alleles, which predisposes towards celiac. So they didn't have full-blown celiac, but there's some sort of connection there. So it was concluded, obviously, that a gluten-free diet can be beneficial to patients who have elevated levels of serum zonulin and do experience adverse reactions to gluten and gluten-related proteins. So serum zonulin is a diagnostic marker, and serum zonulin levels do indicate increased epithelial permeability. There's a plethora of literature on that. And like with any new biomarker, zonulin needed to be compared to what is out there, what's the standards for intestinal permeability. And it was found that levels of serum zonulin were highly correlated with decreased transendothelial electrical resistance, which is what is used in vitro to study the permeability of endothelial cells in culture. Also, the elevated serum zonulin was highly correlated with a high ratio of lactulose to mannitol in the urine after the lactulose mannitol test. So, what are some of the triggers for increasing zonulin and the permeability of the endothelial layer? Obviously, gliadin. Direct adherence, you've heard me say this many times, direct adherence of any bacteria on the endothelial cells. So if you lose those wonderful mucus and mucin barriers, you can have binding of bacteria that will cause a breakdown of those tight junctions between the epithelial cells. other enterotoxins and proteases, corticosteroids, undigested dietary protein fragments, and maybe, just maybe, some food additives like the glue, microbial transglutaminase that's put into ground meat to make it sticky, fructose, emulsifiers, salt, and nanoparticles. And I say maybe because we need more data on that. It's mostly from correlation and some small animal studies. Now, I recently consulted with a case with a doc with a 32-year-old female who had severe lichen planus, bilateral. And you can see a picture of that nasty rash on there. This wasn't actually her. This was taken from the internet. And her initial serum zonulin levels were about 10 points higher than the reference value, so about 55 nanograms per mil. So after 2 months on a product X, which is an intestinal permeability support product, and an effort to decrease gluten, she had a very significant improvement in just 2 months with her lichen planus. I actually saw her, they were just little scars on one side of her body. I couldn't figure out, and I asked a couple of docs, why one side of her body? But interesting. But that was also associated with a decrease in zonulin. So clearly she had a reactivity to gliadin, and when she limited or cut back gliadin, she admitted as a 32-year-old female, she did eat some pizza and drink some beer. Well, have a bagel with that beer, like her at it, come on, man. But it was a significant improvement nonetheless, and that really motivated her to really cut out the gluten. And she's continuing on product X, and we're following her further. Now, a lot of people ask, how does a measurement of serum zonulin compare to the gold standard for increased permeability, the lactulose mannitol test? And I looked at approximately 1,000 samples that were submitted for patients who were suspected for having increased permeability or leaky gut. And we found that there were similar percentage of the patients were high for zonulin at 18% and the high lactulose to mannitol ratio at about 15%. So it looks like they're pretty equal, and with more specimens we'll be able to find out whether zonulin is more reliable or not. But that's the data that I have so far. So, one can use, it's a clinical decision, serum zonulin or the lactulose mannitol load test, but it's a clinical choice, it's a clinical decision. Zonulin is easy because it only requires a non-fasted blood draw, whereas the lactulose mannitol test requires a prescription for the sugars, which is kind of crazy. Dietary restrictions, because there's lactulose and mannitol in a lot of products, and compliance is a huge issue. If a patient misses a urine collection between 2 and 4 hours of that 6-hour collection, all bets are off on the reliability of that test. So the pre-analytical sampling is a big problem with the lactulose mannitol test. Also, it requires giving a 15-gram bolus dose of sugar to patients that may already have GI symptoms. Now, if I had an upset stomach or a gurgling intestine, I wouldn't drink 15 grams of sugar, okay? Again, it's a clinical decision. Now, we know lactulose is not metabolized in circulation in the body, but when it reaches the colon, it's fermented to methane, wind, and acetate. In fact, lactulose is used as an osmotic laxative. It can cause gas and bloating in patients, abdominal cramps, and much less frequently nausea, and very infrequently diarrhea and vomiting. So clinical intervention towards ameliorating that paracellular transport. Number one, just like any other environmental issue, eliminate the triggers. And that's always the most difficult part in your practice. You can start with gluten, okay? And then there's a plethora of literature suggesting different nutrients that can increase the gene expression of the tight junction proteins. And those include specific probiotics that are listed in the reference in the BMC gastroenterology there with the asterisks, prebiotics and butyrate for the reasons I described, glutamine, tryptophan, curcumin, vitamin D and retinol, quercetin, genistein, gamma-linoleic acid, and I hate to say this in front of an AAEM crowd who's been against casein for as long as I've been on the earth, a beta-casein peptide. Don't shoot the messenger, I'm just reporting the facts, okay? Juxtaposed, we know that chitosan, which is ground up crab shells, which is a common food additive, and alcohol, ethanol, decrease the expression of the tight junction proteins. So, ground up crab shells, I'm not sure what they're useful for other than a way to make money with the waste after you take the meat out of the crab. But it's not good for your tight junction proteins. So, my takeaway points are really quite straightforward. The supraendothelial cell barrier is a complex ecosystem of resident symbionts, resident and recruited immune cells, and a very, very responsive mucus layers. The first line mucus layer harbors the commensal bacteria and immune components that fortify the upper barrier layer. The endothelial cell membrane bound mucins, that is the glycocalyx, serve as adhesion sites and protect and facilitate communication with the host through the epithelium. Mucins are dynamic and affected by symbionts and pathogens, and clinical intervention to support mucin should be part of all gut restoration protocols. Zonulin is a reversible physiological regulator of epithelial cell tight junctions and increases the permeability of the endothelial cells. Persistent activation and elevation of zonulin predisposes individuals to inflammatory, autoimmune, and even neoplastic disorders, as well as non-celiac gluten sensitivity. Elucidation of the zonulin pathway has really facilitated development of a definitive diagnostic marker of intestinal permeability to pro-inflammatory and highly antigenic macromolecules from the lumen of the gut. Serum zonulin levels can be measured in a serial manner to monitor your clinical intervention to restore endothelial cell tight junctions, aka leaky gut. And I'm finishing a little bit early here, so I may have time for a question.